go ahead and get started. So it's great to have Neda Jahanshad from USC here. We've talked about collaboration as being really important for how to do data science and neuroimaging. I believe that Neda will talk about a scaled collaboration that's very large, and also it's kind of a very special way of doing collaboration across multiple different sites and multiple different collaborators. And sort of a really interesting model for doing collaboration. And then she'll talk about a bunch of other things as well, I assume. So go ahead.
Thanks so much. Thanks everyone. Thanks guys for having me. This is my first NeuroHackademy, so I'm excited to be here. It's great to meet you all. I changed the title of my talk slightly. You might notice that. So I'm just calling it Continuous Contributions to Large-Scale International Neuroimaging Initiatives. I'm going to cover a lot of stuff. Feel free to interrupt me at any point. Feel free to tell me like something is boring and I'll skip it. Just tell me to go into detail about something. That's totally fine. Basically, I'm hoping this talk can help answer a few questions for you if you're interested in doing research. Maybe you've collected a small data set and maybe you want to contribute it to a consortium. Maybe you don't know of a consortium. Maybe you want to do something else with the data set that you collected. Like, what are the next steps after you're done with the study? What if you want to develop tools and you want to know how to develop maybe some image processing pipelines that can be used widely and applicable for a lot of consortia? Basically, what is a consortium? Why are we talking about this in the first place? What can be done with the data that are contributed to a consortia? And what if you want to lead a project within a consortium? And how do you propose some analyses? And maybe there are some discoveries and findings within consortia that can help you with your research and help propel research forward. So just keep these questions in mind. If I'm not doing a good job of answering them, let me know. And please feel free to interrupt and ask questions. So I'm going to base this talk on the context of the Enigma consortium, but a lot of this is probably applicable to other consortia as well. So Enigma stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. And that entire acronym is really just how the group got started. And we'll talk a little bit about that. But a lot of things are no longer in Enigma necessarily. Like, you don't have to have genetics to be an Enigma. You don't have to do meta-analysis to be an Enigma. And I'll talk a little bit about what all these things mean as we go forward. So first of all, why and how did Enigma even start, and what is Enigma? So I consider Enigma to be a very dynamic and collaborative investigator-driven neuroimaging consortia that's actually organically growing and expanding in both scale and scope. The motivation behind Enigma, you've heard quite a bit about this throughout this workshop already. But really, there's been so much emphasis on reproducibility recently, reliability of results, how a lot of our published findings really don't cut it. Maybe they don't replicate, they don't reproduce. And we've seen how important it is, how many reproducible neuroimaging studies actually need tens of thousands, maybe more, a number of individuals in the study. And there are also requirements for structured analyses and approaches that can help facilitate reproducibility. Now, neuroimaging was not the first to realize this. I don't know if genetics was the first to realize this either, but there was such a lack of reproducibility and false positives in genetics, and this was expensive. There were a lot of costly consequences to these false positive findings in genetic research. So these underpowered studies led to identifying locations in the genome that you think, oh, this is going to be a great drug target for this disease I'm interested in. And then because these are these building blocks that can easily be targeted for treatments, there's a lot of follow-up studies for these genetic findings, but you would have look more into case control associations of certain genetic differences. You'd look at like preclinical mouse models, knockouts. But if these are all underpowered, and we didn't even check the whole genome at this point, there were quite a number of studies that hadn't collected the information on the entire genome. And when that the entire genome was first sequenced in the early 2000s, that's when really this genetic research started taking off, and we started to realize how badly we were doing in genetics research, like how things were not reproducible, how we hadn't screened the whole genome. We didn't even know where we were looking. And then when what was called candidate gene research, which was taking those selective genes and markers that we thought were associated with either with a disease, and we tried to replicate that and didn't find it. We found that just doing an unbiased search of the entire genome through genome-wide association analyses, that showed that those candidates for a lot of disorders didn't hold up. And this is especially relevant for psychiatry and brain disorders. So essentially, genome-wide association studies made the traditional candidate gene studies more obsolete because it was less biased and you're doing an entire search. And the good thing with genetic research is now the cost of genotyping has gone down exponentially. So now you can genotype individuals in your study for maybe less than $15 a sample, where and that helps you get larger and larger sample sizes for studies with the same amount of funding. Unfortunately, the cost of neuroimaging hasn't actually gone down. It might have gone up. And so when we're doing a lot of this work that I'm going to talk about did start trying to combine neuroimaging with genetics. So if we have these genetic findings from, let's say, a screen of depressed individuals versus non-depressed individuals, and you find certain markers in the genome that are related to depression, how does that actually affect the brain signature? How does that change, or how does that relate to differences in brain morphometry, brain connectivity, brain structure? And an example of that, you take a trait derived from neuroimaging, let's say intracranial volume in this cartoon, and you associate that through just a linear regression with the marker and the genotypes of that individual at that particular marker. And then if you do this genome-wide, you're making one association with one trait across all genotypes. And for genome-wide associations, you have roughly 1 million independent variables that you're testing. So in order to establish significance, your p-value, let's say, is should be at or below 5 × 10⁻⁸, and that's 0.05 divided by 1 million. So neuroimaging, we don't have a single trait. So I think everyone here is familiar with the variety of traits that you can extract from images, whether it's at the, just a ICV, a very global measure, whether you're doing voxel-wise predictions or associations, whether you're looking at connectivity, structure or function here. And then the entire genome, you're going to need very large samples. This became a well-recognized problem in genetics and neuroimaging genetics, that the sample sizes really need to be huge in order to get a significance of, let's say, 5 × 10⁸ with one test. So this paper, Statistical Challenges in Big Data Human Neuroimaging, by Steve Smith and Tom Nichols, does outline basically how many subjects you'll need with respect to how many tests you'll need, at the level of variance that you expect some trait will be able to predict. So genetics, especially common genetic variants that you know, they're not rare, but the variant is common in at least 5% of the population. These generally represent less than 0.1% or they explain less than 0.1% of the variance in any complex trait. So this means that essentially for a single trait, a single test, in order to get find an association, significant association that explains 0.1% of the variance, we need roughly 8,000 samples. So this was pretty well known within the imaging and genetics field over the last 10 years. So way back before individual studies like ABCD or UK Biobank existed, probably before a lot of you were born, we didn't have these data sets that had over 10,000 samples. So we had to find a way to make that happen. And we did that by pooling data from smaller studies. So in our very first genome-wide association of imaging traits, we looked at just a single trait, the hippocampal volume, and pooled data from over 17 independent studies. So these were all studies that, for the most part, hadn't really talked to each other before. They were independent PIs from different parts of the world, and they had collected data for their very own specific, to answer their specific hypotheses. And they happened to include neuroimaging and genetics in this work. And these studies ranged in sample size from about 50 to at most about 1,000. ADNI was one of our biggest samples with about 800 individuals in that study. And we also had, we're working with another consortia at the time called CHARGE, and they were also doing the same thing. CHARGE is very focused on cardiovascular health and aging. So they were also very interested in the hippocampus and its relation to dementia. So we had a discovery sample with these 17 studies, and we found one location in the entire genome within or outside of a gene that we hadn't really considered looking into before. It was not a significant candidate for anything, but we did find that it was very much associated with hippocampal volume. And you can see this is a forest plot where we showed the effect and the variance of that effect across each of the different studies that were included. So each line here is a different study, and the width of the line corresponds to the 95% confidence interval for that study. And you can see almost none of these studies could find that effect on their own, even at the 0.5 correction level. So a lot of these studies include 0 within their confidence interval. So they weren't able to find this effect on their own, but pooled and in aggregate across all these studies were able to find a significant effect. So just briefly what was done, each site computed their brain measures, they ran the associations, the regressions, and then the effects were combined. Again, we didn't know at the time whether we could convince people to join the study. Like, why would you want to join the study? Like, you know, why would I contribute my data to you? And this was when data sharing was less common. The study was published in 2012, and the initiative started in 2009. So we said, if you have hippocampal volume extracted, we don't care if you ran FreeSurfer, we don't care if you ran FSL, we don't care if you manually extracted it yourself for your entire data set. If you have that extracted, we're not going to ask you for more work. just run this statistical analysis on your data and let us know what you find. So each site was actually allowed to use any, yes.
Remember how many software were used to assess the sample volume? Because recent works have shown that there was such a big variation in the software, and remember there was like using bias correction for the software.
That's a great question. We had no correction for software at the time. So there were groups that used, most of the groups used either FreeSurfer or FSL. And I think at the time it was like FreeSurfer 5.0 or less. Someone used AdaBoost. This is from the supplementary table right here. A couple of groups had manual segmentations. So there was no consistency. There was no adjustment for software. And again, this was just a very first shot analysis to see whether this concept of being able to pull all this data, these diverse data sets together, whether it could lead to anything. So yeah, there's a lot of variability related to software in this analysis. But what's interesting is that, so this was our first study, we called it Enigma 1. And if you're looking at this plot right here, the x-axis is the position on the chromosome, and the arrow that I pointed to is the single nucleotide polymorphism, or the SNP, that we found was significant in this paper. And the y-axis is the negative log 10 of the p-value. So the higher you go, the more significant. And so the max on this this plot is a p of times 10 to the negative 27, essentially. So we repeated the study again after adding another 5,000 samples after our first publication, and that hit got even more significant. So now we were just shy of the significance threshold. That went well beyond that threshold. And then we added data from the other consortium. We got up to 26,000 samples, and we saw that this finding that we had in 8,000 people was continued to be replicated. And this was a reliable way to find a true effect, because we just kept getting stronger. Not only that, that top hit, if now you look at in the UK Biobank paper, and they've run a GWAS across all neuroimaging traits, we again take that location, and this is, I guess, the VWAS map. Looks like that location is strongest in the left hippocampus as extracted by FAST. And then if you look at all the left hippocampus or the genome-wide association across the left hippocampus, you see that that is, again, the strongest hit. So there was something in that initial publication, by pooling all these small data sets of 50 to 100 to maybe 1,000 samples, that was able to actually get us a very reliable and robust finding of where in the genome we have an association with hippocampal volume. And we're continuing to add more data. So if you, instead of using UK Biobank as a completely independent replication, which this slide demonstrates, we can add UK Biobank into this association as well. And now these findings get even stronger, and we have, I forget the total number of samples here. I think it was over 50,000. We have more, we find more locations on the genome that are associated with hippocampal volume. So these might all be important targets for brain variation. Yes.
I have a question since I'm less familiar with the next step. So you correct the p-value because of multiple comparisons to keep the area at 25. But since you have so many things, is there a chance that something would be stable since you have so many options to be, or opportunity to be stable over repeating analysis, or how would that type of correction work for a effect that could be stable by this? Yeah.
So, so there are different ways to assess replication here. I think we're still correcting for this genome wide, and if you are taking that discovery sample, oftentimes in a replication, you might not even need to reach genome-wide significance because you're saying, okay, I know this one is significant. Does it even reach like 0.05 in this independent sample? But we're actually showing that not only is it significant in just the one trait moving forward way, but it's also significant genome-wide. So it still does reach that significance threshold and actually goes well beyond it, I think. So if 5 times 10 to the -8 is the genome-wide threshold for one test in the UK Biobank alone. And I think this was, I want to say this was like the first 8,000 samples of the UK Biobank. It's looks like 5 times 10, or the P is times 10 to the -13th or something. So it still reaches that threshold. But yeah, that's a good question on how do you actually assess reproducibility. So there's a whole different angle that's not necessarily related to genetics on that. Did I answer your question?
Yeah, I think.
Okay. Any other questions? So now that we have these huge samples, we can actually take our results and make predictions. So we can take genome-wide, a summary score of the genome-wide results and see whether in an independent, completely independent sample, whether the genetics predict volume. And we're not only able to show that so most, unfortunately, still over 90%, I think, of the genome-wide data sets that have genome-wide information and also imaging are largely of people of European ancestry. So a lot of our and other consortia's discovery samples that are used to make the initial findings are exclusively run in individuals of European ancestry. But now that there are other data sets that have genetics and imaging in individuals of non-European ancestry, we're able to see if these genetic findings are predictive in those individuals as well. And we're seeing that not only are we able to make predictions on hippocampal volume in individuals of European ancestry, but also to a lesser degree in people of non-European ancestry. So this continued analysis and keep adding more data not only allows you to make stronger inferences, but take the information you learn and then ask new questions with it. So that's what we can show. And also, now that we have more data, we have more power to test more traits, more phenotypes. So we're not going to just stick to hippocampal volume. Let's run associations with volumes of other structures of the brain. And again, we are seeing that we can predict we can predict some percent of the variance explained in individuals of European ancestry and in individuals of non-European ancestry with these large samples. So this international collaboration is really with people from all over the world. A lot of these people have collected data sets maybe 10, 20 years ago, sort of just forgot about it, stored them for a while. Sometimes they were able to store saliva samples that we help them get genotyped later, because they're saying this will be a well-powered test. It will be part of a larger initiative. So there really is a lot of involvement for a lot of all the work that I'm going to be showing. And now, of course, there are these very large-scale multi-site data sets, which you think that you can do so much in just one data set alone. So these are biobanks, and these large-scale studies are more and more common, and they're tremendous resources. And the good thing is, with these coordinated studies, you can include data from these public resources and combine it with data from private resources for an even stronger, more well-powered study, like you saw with adding of UK Biobank into the genetic analysis. Now, the one thing is, not all data can actually be openly shared, especially if
Jenna, it was one of my references, but she is in Italy right now. So
okay. Thanks, Jenna. So yeah, we can pull all this data together, and we'll have more power for certain studies. But why are we pooling? So if we have all this, we have UK Biobank, we don't, what else do we need? Why pull data from all these small, uncoordinated international sites? Why are we adding variability here? So this is data that a lot of times we paid for with taxes. It's available. Why let it, go to waste? On their own, it might be underpowered for a specific analysis, but that doesn't mean that it can't be useful to help answer questions about the brain, maybe certain disorders. There are specific studies of rare or uncommon conditions or populations of individuals. It'd be great to know if a finding that we have also generalizes to other populations that might be less commonly collected. So, for example, the UK Biobank is quite a bit healthier than the average population. So you don't have, you still have a fair amount of individuals with various neurological and mental disorders, but you might not have as many as you would in a large scale case control study, for example. So a motivation outside of reproducibility and reliability is also generalizability. And we talked about this quite a bit. So we can really assess generalizability in a consortium setting. Not only does a finding hold in different populations, but does it hold with different acquisitions, different methodological considerations, and I think more interestingly, if not, why? And was it something biological? Is it something methodological? Why doesn't a finding replicate? And you can answer these questions when you have a lot of data. So since actually that first publication in 2012, we had a lot of interest saying, hey, this is really nice. You've set up this infrastructure to be able to do this coordinated analyses, like with people doing a similar set of statistical analyses across all these different groups and bringing it together. Why stop at genetics? So in 2012, I believe it was the schizophrenia group that reached out first and said, we want to try this with neuroimaging of schizophrenia. So we started a working group for schizophrenia, bipolar, depression, and now we have over, I've lost count of how many groups we have. There's more than 30. And this is actually an older slide. Just the other day, we submitted a grant to start a group on chronic pain. We have a group that was just started a month ago on language development. So anything really that might be of clinical interest or methodological interest, a collaborative approach to it is feasible, and there are ways which we can go about doing working on that. So there are groups in Enigma that work on algorithmic development, working on, let's say, diffusion protocols that are able to be reliably run across different data sets and maybe applied to a lot of these disorders and these different conditions. We have a resting state group, task-based fMRI group, and a lot of other groups. So if you're interested in something specifically, let me know, and I can tell you if we have it or not. Any questions so far? I hope there are questions. I haven't told you how it works. So we're working with very diverse data. So there's a lot of challenges that come about. So these two images right here, they're actually the same brain, taken on just image taken on two different scanners. This is a 1.5 Tesla scanner. We scanned them just before they had a scanner upgrade to a 3 Tesla scanner. So, but not only are the imaging, the MRI scanners different, the acquisitions different. A lot of times for how we're organizing this consortium, the study designs are different. They're all unique. Everyone was interested in a different population for their own study, for their own grant or pilot project. They have different inclusion-exclusion criteria. They might have used different assessments and different batteries for assessing a wide variety of traits. And to some extent, that's actually kind of okay. So again, harping on that generalizability theme, in the case of finding a good biomarker, it will have fairly limited utility if that biomarker can only be extracted from, let's say, the HCP scanner with the HCP protocol. Nothing wrong with HCP, it's fantastic, it's beautiful, but it might not be something that you can, readily scan on a lot of patients that have certain disorders, and you can't keep them in the scanner that long. So are there features we can extract that are commonly and robustly able to be extracted across different imaging acquisitions? And if people have different criteria for having a case or control around the world, maybe there are differences in how medication is prescribed. Like, it would still be interesting to know if the neuroimaging signatures of those conditions are the same around the world. But we'll get into that a little bit more too. So in terms of the image processing and feature extraction workflows, a lot of the general approach has been around analytical standardization. So Enigma was launched to tackle the reproducibility crisis in neuroimaging, and therefore there's a lot of detailed documentation of the protocols, the analytic plans, and that all becomes actually central to how this consortium works. It goes for the imaging metrics, some of the clinical and biological metrics, the covariates. Something that me and my group have been very involved in is the quality assurance, just making sure whatever protocols gets run, you are visually looking at the data and making sure that it actually makes sense. The statistical workflows, a lot of this gets repeatedly tested, retested, evaluated before it's really distributed on a global scale. So for example, the imaging protocols we check, does it work for multiple imaging acquisitions? Does it work for the data sets we have that are 1.5T? Does it work for the data sets that are 3T? Does it work for kids? Does it work for adults? Is it reliable? And to test that, we often use public resources that have test retest data. So we test reliability within sample across a lot of different samples. Obviously, we want something that's easy to implement. We don't want to give, a few hundred people around the world, a really difficult task that would take them forever to do, and then that would take us a lot of time to troubleshoot. So oftentimes we build our imaging protocols around commonly used existing tools. Either we build approaches on top of it, or we have workflows that work based on their standardized outputs. And most importantly, are the features that we're extracting, like, is there a reason to study them? Are they biologically meaningful? So a lot of the initial Enigma studies were based on extracting free surfer volumes from the subcortical regions and thickness from and surface area from the cortical regions. That was when a lot of people had already run free surfer. We then built a sulcal morphometry pipeline around that. So everyone already had the gray matter ribbon extracted from free surfer that got fed into a new sulcal morphometry pipeline. And before we deployed that, we tested 4 test-retest data sets to make sure that all of the features that we're extracting from the sulcal morphometry are reliable. Not all of them were, so we can focus and narrow down our feature set to the ones that are reliable across these data sets. And we have numerous quality assurance metrics. If you want to look at not only the distribution of the features that you're extracting across your population, but just to make sure it actually did what it was supposed to do. So we have on our Enigma website some tools to help you with visually scrolling through a lot of the data sets. So we have guidelines. For example, here the banks of the superior temporal sulcus are not actually supposed to show on the outer surface of the cortex, and we put a small guideline as to when we would think it's showing too much, and when because it's a very common error in free surfer that the banks actually go on the exterior surface. It's not actually supposed to be there. So we put a little marker, like if you notice that it's bigger than a few millimeters, maybe mark that as an error. And then of course, some regions are easier to identify as not being complete, or just completely missing a chunk of the brain. And once you have had a lot of people go through and manually and visually look at their data, then you can start to build automated tools around some of this. So if you have data sets where a lot of the subjects in certain regions for certain subjects have been marked as QC fail, we can feed that into various machine learning algorithms, which we've been doing on my team as well. And so then these automated tools can help guide QCing in the future, especially as data sets get larger and larger. So sometimes there's new updates with software version, like free surfer was recently updated. And so we have, again, try and test the reliability, reproducibility, and now compatibility with previous versions with actual data. So we have these empirical tests of how reliable and how compatible these might be. This might be important if you're trying to upgrade software within maybe one group, or an entire working group says, okay, everyone changed to the latest version. Let's see how many, how much work we actually want people to do. So a lot of what we're trying to do as the consortium is, because it's so distributed and we have so many people around the world that are putting time and effort into this, we want to minimize the effort that everyone puts in to the extent possible. This is just a close-up of some of the differences between free surfer versions on the same brain. So we've been mostly talking about structural MRI, but actually, as I mentioned, there are a number of protocols being developed for different modalities as well. So we have an active diffusion analysis and quite a bit of resting state and task-based. This is being led by folks in Germany, the fMRIs. So these are continuous evolving trends within the consortium that are often revolve around current standardization efforts, things that certain groups are doing within their own research practice, how it's being propagated through other clinical data sets and clinical researchers, as well as those collecting population level data. So a lot of these standardization efforts that are being discussed here are all actually also accelerating this pace of this multi-site, multi-study type analyses. And as I mentioned, a lot of the tools that are being developed for this Enigma consortium work in particular are being built on existing frameworks. So for example, fMRI prep, there is used as the basis for half-pipe, which is the Enigma version that incorporates a lot of additional output metrics like the connectivity-based outputs and the initial diffusion-based protocols build on TBSS. And there are other versions as well. So a lot of these projects and the protocols are continuously being reevaluated, retested, compared with previous versions, and brought to the consortium setting. So as I mentioned, we can also compare not only these automated volume extraction measures, such as free surfer with the newer deep learning based extraction methods. We can also compare our outputs, whether or not there are changes in the hippocampus with respect to a certain disease, for example, using the ROI based approach and also voxel based analysis. So a lot of these are complementary, and we like to test our findings across different methods. So we know that the finding is not necessarily just due to the particular method that was used. Occasionally, we do have, and more, actually, we have protocols that depend on the data quality. So for example, diffusion, 70% of the data sets in Enigma that were pulled for this table have a B of about 1,000, a B value of about 1,000, and voxel sizes between 2 and 2.5 millimeters. And that's all they have. They don't have high angular resolution. They don't have multi-shell, and they're not optimized for tractography. So in order to develop a tractography protocol that is optimized to have the most connections possible, we need to sort of tailor it to what the consortium has. So for example, there are a few data sets that have larger than 2.5 millimeter voxels. If you work with diffusion, you know that that's not the best to work with. And maybe they have about 12 directions. So really doing tractography and something like that is limiting a lot of the other data sets to the lowest common denominator, and that might be too low. So we oftentimes put some filter to say, okay, you need to have at least this, this level of data quality to be able to run this protocol. And that allows allows a lot of the newer data sets that have been collected or allows the consortium to make the most of the newer data sets that have been collected with the better resolution and the better acquisition protocols. So all the standardization with these protocols, extracting features a certain way, the documentation, containerization of protocols, it not only allows groups within a working group that we discussed that might be, interested in, let's say, purely schizophrenia or Parkinson's, to work within that working group, but also you can look at common effects across these working groups. So we can now see on average how the effect sizes for schizophrenia differ from the effect sizes in bipolar disorder across the entire brain in these very high-powered studies, all of which that had over a couple thousand cases and a couple thousand controls. Yes.
Never had the case of so much data that effect size is like a significance is really everywhere.
What do you mean?
I mean, even a very small effect size that doesn't mean biology it actually is significant.
Yeah, so actually a lot of these effect sizes are very small. And that's what we're finding is a lot, and I think that's part of what Enigma's brought to the table, is that a lot of the studies that were showing huge effect sizes between people with depression and controls, those were very biased, and it's actually not that strong. I think the effect sizes we were finding for major depression were around the order of like Cohen's d of 0.15 or 0.2 at max. So these are very subtle effects that we're working with, and that's, I think, why the Enigma's taken has had such a role in psychiatry, is because a lot of these neuroanatomical effects have been so subtle, at least with brain structure so far. So now there's a lot more work being done in function, microstructure, and connectivity. Any other questions? Yeah, so I was hoping someone would ask about harmonization of features. I know you were all thinking of it. So I actually left that out. So we didn't even talk about how we're harmonizing across features. And that's actually because a lot of the work that I've discussed so far, the level of data sharing has not been on the individual level. So a lot of the work has been distributed where the individual sites are doing all the analysis, they're running the stats on their end, and then they're sharing only statistical summaries. So that's because a lot of this data cannot be publicly shared or openly shared. For those of you from the EU, you know that the GDPR has really put a damper on a lot of collaborative work, but we can still work around it. We can still, we work a lot with Europeans, and there's all over Europe, we have sites and people leading projects. So a lot of this has been framed in what we call a meta-analysis framework, which we'll talk about in a second. So sometimes a lot of things, certain data aspects are shared. Raw images are not often shared very often. Of course, there are privacy concerns. And we make use of this distributed framework. So even though data can't be shared, open science this is still very much an open science framework. So I really like this definition of open science that was published in 2018. It's sort of like combination of different definitions that had been that had been previously defined for open science, but basically they say it's a transparent and accessible knowledge that is shared and developed through collaborative networks. So there's a lot of aspects of sharing, but also developing sort of this within a developing methods and protocols through collaborations, transparency, such as pre-registration, peer review, and accessibility. So making sure, even if the data might not be accessible, information about the data is, or the findings are. So Enigma is actually not a data repository. So data from the participating sites, as I mentioned, there's a lot of different data sharing rules that we work with, a lot of access rules, a lot of the data sets that we incorporate in these studies are public, so those are free. A lot of them have very strict restrictions, especially if they're of patient populations. That makes it very sensitive sometimes. But any working group member that might be involved in a particular working group can propose a project. So these projects are often first go through the chairs of the working group, but they just make sure that there is sufficient interest, there might be data that's not overlapping with existing projects, and then they present it to members who have their own data, and they can choose to participate in that project, or they can choose to opt out and not be involved. And then the leader of the project will work on how to access the data if data access is needed, and how the analytic plans are done. Maybe taking a little bit of a step back and looking at the working groups, and these are often, again, revolving around certain traits or clinical conditions of interest. For example, the Parkinson's working group, we have a working group on suicidal thoughts and behaviors. There might be certain data sets or certain cohorts that are involved in multiple working groups. So when a working group first starts, there's often an initial project that is led by the working group leaders. And the main, they really try and the initial projects try and ask very straightforward and broad questions that will let as many sites join as possible. So oftentimes these are, I don't want to say simply, but simply case control studies. So just cases versus controls across all the data sets, maybe covariant for very few variables that all data studies have collected, like age, sex. And so this just really sets the stage for being able to ask more nuanced questions and finding out exactly how all the different studies were conducted, what additional variables they might have collected, whether, let's say, it was certain cognitive traits and certain neuropsychiatric batteries. What is it that we can really bring together across all these different sites? And maybe it's not all sites, but maybe we have to filter. So those initial projects that are very broad, those kind of help determine the scope and feasibility of future projects. And so within each working group, we really try we're trying to structure a lot of the data types. So this is a lot more challenging than trying to work with the neuroimaging, is to try and figure out the types of data that each group has collected. Let's say you have 20 different studies all studying Parkinson's involved in this consortium, but each group might have been interested in some different aspect of Parkinson's. Some groups might have been interested in cognitive impairment in Parkinson's. Other groups might have been interested in sleep disturbances in Parkinson's. But now they're all coming together to look at neuroimaging differences between within Parkinson's. So if you want to propose a specific project, you might have to filter through some of the studies within the working group to see who can join your project. So not all groups can join every single project, and that's okay. But oftentimes we do need to do more, ask more nuanced questions to really understand specific aspects of the disease. And these project proposals, in a sense, also act as a sort of pre-registered analytic plan, because the proposal, the person who's proposing the proposal sort of writes up what they're interested in, the background, the methods that they'll use, and then they distribute it to the working group members, who later decide whether they can and will join or not. I know you saw this from Professor Gerton a couple of days ago, but even there are these two paths towards this robust understanding of outcomes within consortia too. So we can have these like very general questions that we're asking, but also across multiple data sites or data sets within a consortia, they can also be very focused. And that's becoming more and more the case within Enigma, when we're looking at certain groups that might have outcome measures and longitudinal data. So this is something we're actively working on now. So the meta-analysis concept that I brought up before, those are projects that only required summary statistics from each cohort. So it's basically the overall group effect size is just a weighted average of that of the individual cohorts, and usually are weighted inversely by the variance of the finding. These coordinated meta-analyses are a little bit different than the literature-based meta-analyses that you might be more familiar with. And they're not subject to publication bias, because everyone is running the same protocols. You're getting the information from all of the tests that you run, regardless of whether it's significant or not. And it's easy to see across these sites when you create these forest plots, the heterogeneity in the estimates and also any outliers. So this is an actual example of something we did in the diffusion analysis of schizophrenia. We did case control analyses. This is, I think, the FA, the average FA in the white matter across different cohorts. If you can sort of see the vertical line indicating a zero effect is somewhere here. Almost every single cohort is showing a negative effect, except this one, which is saying the effect is positive. So that looked very suspicious. And we went back to the site and said, can you check a couple things for us, please? And it actually turned out that they had miscoded some cases and controls. So this, doing a meta-analysis this way and kind of comparing effects and not only effect sizes, but the directionality of the effects kind of gives us some insight, yes.
For the meta-analysis, everyone is the same processing pipeline.
So that's a good question. A lot of Enigma groups are working towards that. A lot of, in this particular case, the pre-processing was different. But the post-processing of the FA values themselves from diffusion was the same. So anyone in this particular case was allowed, again, because we're trying to minimize the work on the researchers' end. If they had already, extracted FA maps from their diffusion, they were able to use that and just run a minimal set of protocols to get the exact regions that we were interested in. But now there are definitely a lot of efforts going towards harmonizing the pre-processing as well. And it'd be interesting to see how much of that variability is reduced by sort of harmonizing the pre-processing. Well, we actually do have guidelines for people that have not pre-processed their data already. So we do expect to some degree that the people that have worked with their data are more familiar with it. And because there's so many types of data sets out there, I can't claim to be an expert on someone else's data set, right? So to some degree, like we will hope that if they processed their data, they knew what they were doing, right? So they tried a bunch of different tools, and then they did the pre-processing that was best for them at the time that they did it. So, and it, for the most part, has worked so far. A lot of times, if they haven't done any pre-processing or there isn't a neuroimaging expert on their team, we're happy to help guide and then have them remotely do processing or give them the tools to do it. So. Yeah, so one of the drawbacks of meta-analysis, unfortunately, is that it's really limited to associations with common traits. So if you have genetics across your entire population, we're only looking at common genetic factors. And with a lot of the case control work, you really have to have a good distribution of both cases and controls at each individual site. So that's sort of one of the limitations. It does take a long time to troubleshoot, sort of in relation to your question. If people had not done processing, you're essentially working with each individual site to make sure they're doing things the right way. You can look, at different stages and help them with quality control, and sometimes that is the last stage. So this is, I won't get into this, but we did create this sort of distributed analytic framework. The genetic projects, as I mentioned, they have all been meta-analysis, and again, this is where the name came from. It started with imaging genetics and started with meta-analysis. So each site does the feature extraction and the statistical analysis with some protocols that we set up, or the analysis methods, we have some scripts, usually in R, that people will run. Everything is on the GitHub. And then the feature extraction, as I mentioned, we have these tools for people to work with if they haven't already processed their data in the right way. And then the summary statistics come back. They're all at a central location. This is for one of the latest genome-wide associations for the cortical structure. And then we do site-level QC, make sure everyone's results make sense. It's the null is as expected, and then we look across data sets and look for trends and strange outliers and variability and effects. A lot of the work in the disorder-based working groups have also been with meta-analysis. And there's a lot you can do with meta-analysis. So we've looked at cortical abnormalities, white matter microstructure differences, subcortical differences between cases and controls, and then also looking at, let's say, white matter and cognitive ability. So all these were in a meta-analysis framework. Like asymmetries between left and right in meta-analysis and brain age. So we had a common brain age distributed protocol for everyone to use. But now a lot of projects within Enigma, as data sharing is becoming more recognized as a necessity, there are a lot more projects that are being proposed that you could just call it a data analysis, but to contrast it from Mega, we're putting Mega. So a lot of, so in this case, it refers to analyzing all the data from really a central location that has pooled all the subject level variability. And as opposed to meta-analysis. So I put an asterisk on centrally because there's also a lot of efforts. I don't know if anyone is familiar with some federated analyses and approaches, especially some federated machine learning approaches. So there are some tools being developed where you don't actually have to share the data, but you can still run analyses across all subjects. So often the mega-analysis approaches use features that the sites have extracted already. So they might run FreeSurfer and then send the individual level free surfer outputs, as opposed to running free surfer, running stats, and then sending the stats. One of the advantages of this, obviously, there's less back and forth with the participating sites a lot of times. Sometimes they are not interested in running the image processing, and they're like, here's the raw data, please just let's work through MTAs and you do all the processing. But a big positive is that you can assess rare effects. And I'll show some examples of some rare effects that we are looking at. And then you can essentially run analyses on traits that are maybe not well distributed across sites. And that could be case control. You could also do like a lot of machine learning now that you have all these data sets. A negative is that site effects do need to be taken into consideration, and you do have this harmonization. A lot of those protocol variations are actually taken into account in the variance when you're doing a meta-analysis. So that counts as part of the weight. So if you have, let's say, hippocampal extraction protocol that's known to be very variable, it has a lot of noise, your statistical results using that method will also have very wide confidence. So that doesn't get weighted as high as something that's more precise. That's not true in the case of mega-analysis, because you're pooling everyone together. And I always like to recommend, if someone is doing a mega-analysis, they still do the site-wise analyses, just to see if there are some outliers like we saw in this example right here. So there's a lot of exciting developments in statistical data harmonization, a lot in the combat family of harmonization. I'm not going to go through all these. I think there's a lot of great work being done, and they keep adding new tools. But this is a very active field of research, and sorry?
Do you have any papers on
Diffusion, what level of for tractography? Tractography is a very challenging one. We do have actually an open challenge at CDMRI this year, as part of MACAI, it stands for Computational Diffusion MRI workshop, as part of MACAI, that is actually asking just that, like, can someone come up with good diffusion harmonization tools? So there are different levels of harmonization. You can harmonize the raw image. So there are some papers that have been published for that. And then you can harmonize also the extracted features. And tractography is a big challenge, but things like regional microstructure are very similar to if you were to harmonize, like hippocampal volume or anything. But so a lot of work so far has used those similar statistical harmonization approaches, like combat for like regional FA for diffusion. But yeah, a lot of tractography harmonization also depends on all these protocol differences. So depending on the level of harmonization you want and the data sets that you have, I think Ariel and Noah White might be better to answer a lot of those questions too. I don't actually know all the work that's being, there's a lot. There's a lot of, I think it's a very active area of research. So in terms of the combat family, we've done a couple or empirical tests of different combat approaches and how it works with some Enigma data. In the PTSD working group, we looked at harmonization. Sorry, I think I skipped one, but so we looked at harmonization using combat, not using combat, and we saw combat GAM, the generalized additive model showed somewhat of an improvement over the others. This was also the case. This is for global fractional anisotropy from DTI. The combat GAM seemed to preserve the curves of the lifespan a little bit better. So with data harmonization and mega-analyses and sort of controlling for the site effects, you're able to look a lot further into the data than with the meta-analysis. So, for example, in our Parkinson's disease working group, we're not only able to look at case control effects across these studies, but after we make adjustments for site, we can also pull together people from or people at different stages of the disorder and really try and look at the progression of changes in neuroimaging signatures across these disease stages for severity. And there's a lot of times where certain sites have not collected individuals that are at all disease stages. So you can't necessarily do this within a site, but only when you pull the data together do you really have the power to break this up. Another working group I'm very involved in is the Enigma Suicidal Thoughts and Behaviors group. So we're actually, for this group, and this is, you don't necessarily have to be able to read this table, but for this group, this is a transdiagnostic working group where we're looking across a lot of different mental health conditions and pulling to see if anyone has collected information on whether someone's had a suicide attempt or not. So suicide attempt has a very low base rate, and to be having necessarily scanned someone that has had an attempt is going to be relatively rare. So a lot of our cohorts almost all have 10 or less cases of a previous attempt. And so the only way to really do a well-powered analysis is to pool all this data together rather than to be able to do individual site analyses, because with an N of 10 or N of 8, even 7, it makes it not something that is well powered within the individual sites, and the noise will be just too much. So even within this group, though, we can, and we've had one published paper where we looked at youth only, and we're able to narrow down our hypotheses as well, look only in individuals with mood disorders and that have very detailed assessments of suicidal thoughts and behaviors, and then more broadly expand. So we took actually a 3-stage approach here, a very focused approach first, and then we added more variability to the samples that we were testing, including adding individuals of cohorts that had different cases. That's instead of mood disorders, we also had folks with OCD, for example. And interestingly enough, our effect was actually limited to the more narrower sample. But there is one strong limitation of statistical data harmonization methods. It's that if your site differences are too vast, or if they're confounded, you might not be correcting for site, you might be correcting for something that you want to preserve instead. For example, we had this recent study that we published of brain alterations in individuals living with HIV, and as associated with the viral load of the individual, and also just immunosuppression or CD4 counts. And interestingly enough, like nearly 80% of our cohorts across all these cohorts are male. So we wanted to look at male and female separate in this cohort and look at the effects of immunosuppression in females with HIV. And it happened to be that the group that there's one group that was predominantly female, and that was one group that was also predominantly untreated. So these were people that had very high viral loads, very low CD4, and they happened to be almost exclusively female. So, so if we should do any statistically based correction, you don't know if you're correcting for sex, you don't know if you're correcting for site, you don't know if you're correcting for viral load, because this one site was a very extreme, and I think something like 75% of the females that we had in the sample were from that site. So it's just something that is really was brought to light when doing this, is that we can't do a statistical correction and completely rely on the results. And we still did male and female separate, but there is this obviously caveat that it's very confounded. So we need more active research and harmonization methods. And this is something we've been trying to work on in my team and others. One example that was just published a few weeks ago online is this method with style encoding adversarial models, generative adversarial models, where we're trying to match the style of another MRI scanner and recreate the raw image of that individual as though they were scanned on a different scanner with a different protocol. So there are extensions of this work that are being conducted. For diffusion, that I wanted to put a little plug out here for this Mackay diffusion harmonization challenge. So this challenge should, when we test the results, we're testing on how well microstructure was harmonized, how well tractography was harmonized, and connectomics measures were harmonized. We have 100 subjects that were acquired with 2 completely different diffusion protocols at the same session. For those of you familiar with diffusion acquisitions, one has, this is all in 4T, so it's a very unusual data set. One has 27 independent directions and 5.5 millimeter voxels. Nightmare. And the other same people is 105 or 94 directions and 2 millimeter voxels, 2 millimeter cube voxels. So how are you going to harmonize those data sets together? So I think submissions are due September 8th. So I just wanted to also mention another approach towards harmonization is creating, in a sense, harmonization is creating these lifestyle charts. I'm sure you've seen this really nice paper from the Brain Charts Consortium where you can map these normal trajectories for different brain regions and look at deviations for an individual with these normal trajectories and whether these deviations can be used as biomarkers. You can look at differences in the peaks of maturation. So we've actually, this had been and still is an active area of research within the Enigma Consortium as well. And we've recently created this website, centilebrain.org, where we tested a number of methods and created this this reference that if you go and you upload your data, we will give you deviation scores for each of your individuals with respect to this normative curve. There's a lot of data that went into this these curves. I'm not going to get into that, but we did test a lot of algorithms for how this harmonization should be conducted. If you're interested, this preprint is on bioRxiv. So we still need more data. So I know there's a lot of data sets out there, and we're still, we have so much resources, but really, even a small sample study can be aggregated with larger studies, and you can look at new populations, new technologies, and advanced acquisition method, or just nothing new, really. You can just use it to validate findings, refine conclusions, and increase your confidence in existing results, and just make more use of the data. And just yeah, I'll kind of just gloss over the rest, but we're also trying to create, still under development, an infrastructure where you can actually add to existing studies. So in this GWAS of the hippocampal volume, we noticed that ApoE, many of you will be familiar with the ApoE genotype. It's one of the biggest risk factors for Alzheimer's disease and dementias, and it's one of the only genetic variants or common genetic variants that is substantially, I'd say, associated with a neurological or psychiatric disorder or complex neurological and psychiatric disorder. So interestingly, though, ApoE has been repeatedly shown to be associated with hippocampal volume. In our GWAS, it wasn't. So, or it was just sub-threshold, actually. So the p-value was not times 10 to the -8th, but times 10 to the -5th. So we're like, well, why is that? We wanted to look into that further. So by having all these data sets, and not even the individual level data, but the data set results, we're able to see that it's actually the older you get, the older the data set average age was, the more associated it was with ApoE. So ApoE might not have an effect in youth, but it does in later adulthood. So that's what we can take our findings and propose new hypotheses with them. So we can take the results and the data and ask further questions. And that's one example of what this is intended to show. So this really just is sort of a cycle. We ask the questions, we design the approach, put more data in, we can keep conducting new analyses, generate new findings. You can keep that initial GWAS going, but then maybe there are results later that you want to look at. So essentially just comes this like living, breathing thing with a lot of like sub branches of different studies, and you can pull it all together later. So kind of thought of the circulatory system and the merging and the sort of dividing of the different branches. I'm not going to get into the infrastructure that we're trying to develop for that, but I did want to point you to a few resources that you might be of interest or might be of interest to you. A lot of these are on our Enigma Git, the GitHub for Enigma. There's Centile Brain, which I mentioned. You can go through a lot of the results, the published results of these case-controlled differences in the studies. If you are interested in knowing what the effects were for different regions for a particular disorder. We have tools within the Enigma toolbox that will allow you to do that. Enigma Viz is for the genetic results. And there's also a new initiative called Enigma U. This is generally targeted towards undergrads. It's a free online set of courses targeted towards neuroscience and neuroimaging, and introducing folks to that. And thank you. questions. Yes.
Yeah, I'm not sure if I understood correctly, but you were mentioning that potentially correcting for side effects might, obscure some actual differences that might be going on. So in your case, in your experience, like, are there any things we should control for that people are always important, or is there like a feature that we should maybe do it with controlling, without controlling? Or in your case, like separating and also like that.
Yeah, so sorry.
Do you mind repeating it?
Oh, sorry. So the question was, because statistical harmonization can have varying results and sort of be driven by all the different variables that you're including and the variables specific to the data sets. How do you know what to control for across everyone and what not to control for? And I think the answer to that question is generally in the unknowns. A lot of times you don't know what's been what has been collected in that data set. So you can ask for the things that are commonly collected, like you can control for age, you can control for sex, and then hopefully you preserve the variabilities there. But if you don't know if those are confounded with other things, I think that's where the problem lies, is if you don't know that, like in our example, the essentially only largely female site was also the only one that was had untreated HIV. I think it's the problem lies more in what you don't know than what you do know. So you can control for everything you do know, but then there's always a risk that there's something that you forgot to ask for, or there's something specific to the data set. So that's why I actually think having this collaborative approach is a bit less risky, because you can just ask the investigators, like, what was your study design? Tell me, like, what else did you collect? Like, was it specific to something? Whereas you might be less familiar with, a data set that was made public, but you don't know all the variables that they might have collected, or, a lot of the reasons behind the study. So I think having the collaboration does allow you to kind of open a line of communication and learn more about the data set. Yeah, I'll always check. So a lot of times we do these like leave one out analyses or leave multiple data set out analyses, and just to be sure that things aren't being driven by a single cohort. Any other question? Yeah. Yep. Yes.
Oh, sorry. What are some of the challenges with, like, keeping researchers engaged for this long term? Like, I imagine folks who from institution to institution, they're not in charge of that data anymore. The data is old. Like, how do you people?
Yeah, so that's a great question. How do you keep people engaged, and how do you deal with people leaving institutions and leaving data sets? It's a challenge, that's for sure. A lot of times, if people are very involved in Enigma, like they are involved in the publications, they're involved in the analysis, they do put effort in. So there is some ownership to the findings and the working group from all members, really. A lot of times, if people do change institutions and they don't have access to the data, they do put someone in charge. They're saying, okay, this person will represent this data set for Enigma from now on, because there has been that relationship that's been built, and there has been, I guess, some level of trust and interest. But it's just a challenge. Yes.
Question regarding, so you mentioned that the data you receive is usually pre-processed by the team in the pipelines that they're used to, and if not, you'll point them in the direction that you kind of prefer, so we give a little bit of nudging. So if you, in the context of harmonization, because obviously by the points we provide, it's essential in some cases, despite the biases mentioned. If you provided the group's containers to use to pre-process and extract parameters that then you use, do you feel as though harmonization is less necessary or still?
That's a great question. So the question was, if a lot of times people are pre-processing the data on their own and then giving results, and then how does that compare to containerizing protocols and having everyone pre-process the data from scratch. So there have been efforts in Enigma to do that, actually, to just start from scratch. And this happened really again after the initial trust, and everyone was like, oh, okay, we're getting more out of this. We're getting a lot of papers out of this than we're putting time into. So yeah, so we'll put in extra time and we will process our data. Because so much of the data has been collected across so many scanners, across such a wide variety of acquisition protocols, there still is a need to harmonize. A lot of the pre-processing tools, you have to have specific flags that you set for a particular data type. For diffusion, for example, if you've collected APPA, you process it one way. If you didn't, you process it another way. So it's not 100% the same regardless for the most part. FreeSurfer is easy because everyone has structural MRI, and for the most part, they're like 1 millimeter cube voxels. If you go beyond that, it's a little bit more challenging. And yeah, I think harmonization is still necessary. But the amount of variability that gets explained, I don't know. We haven't empirically tested that. Yeah, JB.
I'm not sure if we could move to a more prediction framework. But yeah, you develop things from end side, so you predict the other ones. Because that's to some extent some of these musicians.
Yeah, so that's so in terms of predicting outcomes, that's actually something we're trying to do in a lot of these working groups is, can we actually predict from the imaging case versus control, for example. I don't know if anyone here has been involved in some of these prediction challenges. I think we had something at OHBM 2019, I want to say. And for a lot, or for at least for depression, I think the accuracy in prediction is roughly like 60%. So it's not that great. Neuroimaging alone, I think, can help a lot of these, but there are, at least that's for depression, which we saw the effect sizes were really small. For a lot of the other disorders, there have been higher prediction accuracy, but we are working towards not just predicting case versus control, but trying to discriminate between disorders, responders versus non-responders for certain medications. The epilepsy group has an active area of research on that. But yeah, we're moving towards more interesting questions. Yes.
In light of the new harmonization models at risk, is it better to use this sort of like meta-analysis for to like reprocess and republish the more foundational papers that were like collected and established with smaller subsets, or is there a group that is more looking for it?
Yeah, that's a great question. Whether mega-analysis across these data sets with harmonized tools, whether we should go back and redo the original associations, and that was your question, right?
The foundational.
Yeah, some of the so that's a really good question. I think that is to some extent what we've tried, what some of our initial work has been is, there for a lot of the for some of these disorders, at least for a lot of the psychiatric disorders, there hasn't been a 100% well-established effect size across the brain, like a map of the effect size across the brain wasn't established before. So sure, there might have been certain regions that were thought to be foundational to that disorder, but brain-wide, there was less information. And so I think we tried to kind of take a page out of the genetics book rather than kind of focusing always on that one region, do the brain-wide analyses and kind of add to the knowledge. And the fact that we found some of those key candidate regions says neuroimaging is better than genetics in the first place. Was that recorded? But we're still adding to the knowledge. And whether the harmonization and site controlling adds beyond the meta-analysis, we've had a couple publications that tried to empirically actually answer that question. They compared the traditional meta-analysis approach we took and then tried mega-analyses a couple different ways with statistical harmonization, found just marginally better, stronger effects with harmonization, but nothing too different. Yes.
Do you have any working groups or do you know of any similar groups that are using pet data in this way?
So not yet. I think there's a lot of efforts in the Alzheimer's community trying to harmonize pet data. So not within Enigma, we don't have that, but I know there are other efforts. But yeah, that's a great question. Any other questions?
Thanks, Neda.
Thank you.